So let's get started here. My clock says 8:30. So it's a pleasure to introduce Coco Ugbobuaku for her thesis seminar. She came to us from the University of Lagos, where she worked on population genetics of Drosophila using chromosomal squashes. So if anyone here has done chromosomal squashes, you know that anything you do after that is going to be much, much easier. So I'm excited for you guys to hear about her work, which she has really led sort of an aspect of our research focusing on sort of really local population differentiation of behaviors from areas, around Clarksville and Montgomery County and slightly farther afield. So really sort of continuing some of the themes that she did in her undergraduate research, and I'm excited for you guys to hear about it. So with that, I'm going to pass it off to Coco.
Hi everyone. So today I'll be talking about the effect of being in a densely populated environment and the cost that it confers on females during reproduction. And also I'll also be talking about the role of maternal effects on competitive abilities. So, intraspecific variation. We see variations amongst individuals of the same species, and we want to ask why. That is, why do we have a frog that has an orange color and a frog that has a green color of the same species? Over the years, so many scientists have delved into uncovering the factors that leads to this variation. Where does it come from? Some of this variation can come from, one, plasticity, in the sense that organisms constantly monitor their environment and they detect changes in this environment, which can influence they detect changes in this environment, and then this influences a response to the environmental changes in this environment. And this response allows them to cope with these fluctuating environmental conditions, thereby leading to these variations we see in individuals of the same species. Another possible way in which interspecific variations can exist is local adaptation. Now, this occurs when one species diversify into different environments, and then those environments select for traits that allows them to thrive in such environments. So as a result of this, populations can be genetically differentiated, with each of them adapted to their own specific niche. Now, there are so many factors in the environment that can confer this response that we see. One of these factors is competition, which could be as a result of high population density. Competition, which could be as a result of high population density. Now, living organisms in their environment compete for food, they compete for mates, they compete for space, and even competition for reproductive resources. And intraspecific competition is the most intense form of competition, and then it can manifest in various behaviors like direct aggression. It can manifest in social dominance or indirect methods like reproductive suppression. And also these behaviors can influence various life history strategies like reproduction. Now it influences reproduction in the sense that it can shape reproductive strategies and influence reproductive decision making. And this can invariably influence variation in body size. Also, it can also influence an individual's survival, which I'll get into a little bit later. So let's talk about how competition influences reproduction. Competition can shape reproductive strategy in the sense that in an environment where the food resources is scarce, a female will develop strategies in order to maximize these resources so that she can ensure greater reproductive success. Also, it can also influence our reproductive decision making, especially in individuals that provide parental care to their young ones. Decisions like how much energy should an individual invest in a particular round of reproduction. And this is the basis for the cost of reproduction hypothesis. What is the cost of reproduction hypothesis? It posits that there are trade-offs between current reproduction and future survival. In other words, this means that investing energy and resources to reproduction might reduce an organism's ability to allocate resources to other vital functions such as growth, future reproductions, or maintenance. And this can also present a trade-off in the number and size of offspring produced. Now, how does density or how does competition influence body size? In an environment where there is fierce competition, an organism or an individual would have to develop certain adaptation to be able to survive in that environment. Adaptations like bigger body size, modification in their mandibles, in their beak size, or their limb. A study by scholar Price and Grant et al. They did a study on Darwin's finches. You can see that these Darwin's finches possess like variation in their beak size. So they attributed this variation to competition for resources, for food resources, which came about from drought in their environment. So other studies have shown that in a competitive encounter where size is the limiting factor, bigger individuals mostly win or always win, and body size can also reflect the intensity of competition in an environment. Curtis Creighton did a particular study where he highlighted that individuals or reproductive output in a competitive environment was characterized by larger body size and smaller clutch size. Thereby, I would assume that the essence of having a bigger body size in a competitive environment would be to confer this competitive advantage and also ensure the survival of the individuals. Now, how does competition influence survival? Competition can have direct effect, both direct effect and indirect effect on survival. In direct effect in the sense that if a competitive encounter should lead to aggression, a fight can break out, and this can lead to injury or death of the individuals involved. Also, in an environment where there is starvation, where there is competition for food resources, and an individual cannot get that food resources, they can die from starvation. Competition can indirectly affect survival in the sense that higher reproductive outputs in a competitive environment can be very energetically costly, which can lead to reduced fecundity. And then a decrease in fecundity could be a sign of senescence. And we know that senescence is like the gradual cellular death. So all of these are ways in which competition can actually affect various life history traits. So for this thesis, I aim to explore how environmental variation can induce changes in reproductive behavior and phenotypes, and also understand the benefits of this variation in reproductive tactics. And lastly, I want to understand the mechanism that underlines competitive interactions. So I used the bearing beetles Nicrophorus orbicollis for my focal species. They belong to the family Syphilidae, and they are carrion beetles because they feed on dead animals' carcass. Why did I use Nicrophorus orbicollis? These beetles have been largely studied because of their life history trait, but I used them because I was interested in they exhibit variability in body size, like high variability in body size, and they have a very interesting reproductive behavior and also intraspecific competition dynamics. Now let's talk about bearing beetles' reproductive behavior. These beetles are one of the species that exhibit parental care to their young ones. Now they have this chemosensor in their antenna in which they use to detect environmental cues and and also detect the presence of carcass in the environment. So when they detect this carcass, they prepare it using anal and oral secretion. They do this so that they can prevent this carcass from decaying and also prevent other competitors from perceiving this carcass and stealing it. So when they do that, they bury it in the soil and then they lay their eggs. So when their larvae emerge, they feed their offspring mouth to mouth. So this is a video of a beetle feeding its offspring mouth to mouth. I know the video is kind of weird because it's red, but these beetles are sensitive to white light, so the only way we could carry out observation was under red light. So they feed their offspring mouth to mouth, and in a situation whereby, or in an environment whereby there is like limited resources for reproduction, they can engage in like infanticide, like killing some of their offspring. So all of these have to do with the decision that they make in the environment. So this parental care behavior allows us to study the effect of densely populated environments on reproduction. So now let's talk about beetles' body size. Bearing beetles exhibit high variability in body size, and this variation can exist among and between population. A study by Son et al. Showed how the beetles from two different populations that are like 1.2 kilometers apart, like in this map, they were like 1.2 kilometers apart, but then they varied in body size. The body size variation was really high, and they attributed these differences to the density of the environment. So this means that body size can actually reflect the intensity of competition in an environment. So why am I doing this study? So there have actually been studies by Curtis Creighton and, like I mentioned, Son et al., which have told us that there is a trade-off in a densely populated environment in the number and size of offspring that an individual produce. But I want to uncover the effect of this trade-off, that the cost that this trade-off presents on the females in reproduction. And I also want to understand if maternal effect plays a role in competitive abilities. So I carried out two studies. The first one was to investigate the influence of density on reproduction and reproductive decision making. And the second one was to investigate intraspecific competition dynamics between two distinct populations of Nicrophorus orbicollis. So let's talk about the first study. Effect of density on reproduction. Reproduction as a life history trait comes at a cost, depending on the factors that affect it. So here we want to examine how population density affects reproductive output and lifetime investment in parental care. So why this study? Like I said earlier, bearing beetles exhibit variability or high variability in body size. So during my pilot study, I observed that beetles gotten from 5 different locations. These 5 different locations, I know we can't really see the vegetations here, but beetles gotten from these 5 different locations, I observed that they exhibited like variabilities in body size. Like a particular population had dominance of smaller body size, and then a particular population had dominance of bigger body size. And from previous studies, I've seen that, from previous studies like that of Creighton's, like I've seen that body size reflects how dense an environment is. So this led me to ask questions about like these populations that have this smaller body size. Can they exhibit plastic response if they are exposed to a high density environment? or have they lost plasticity, or this population that exhibits bigger body size, have there been like local adaptation? Have they been adapted to their environment, their competitive environment, and have there been local adaptation? And also, what is the cost of reproducing in a densely populated environment? So I developed two hypotheses from my questions. So the first one is that in a densely populated area where resources is limited, a female will prioritize her current offspring over future reproduction. And then this would come at a fitness cost to the females. So I expect to see a reduced weight gain in females that are in a high density environment after reproduction. Also, because I know that a trade-off associated with the high density environment is smaller clutch size and bigger body size, I expect populations that are characterized with dominance of smaller body size to not exhibit any plastic response when they're exposed to density treatment. So my second hypothesis is that in a high density environment, will act as a catalyst to speed up the rate of senescence in a reproductive active females. So if this is the case, I expect to see females in a high density environment experiencing faster mortality. So how did we test this? Or how did I test this? First of all, people from my lab and I got beetles from different locations in Tennessee, and then we bred them under the same laboratory conditions because we wanted to get the F1 generation. So we used the F1 for this study. So after they became adults, we exposed them to two different density treatments for 2 weeks. The first one is the high density treatment, and the second one is the low density treatment. The high density treatment, this is an example of the high density treatment box. It contained 10 beetles in the box, while the low density treatment contained 1 beetle in the box. And this picture is just showing that we fed them equal amounts of beef like 2 times a week. So we allowed them to attain sexual maturity in this box because beetles takes 2 weeks to reach sexual maturity. So we exposed them for 2 weeks before we proceeded to breeding them. So before we bred these beetles, before we bred the females, we weighed them. And after breeding, we also weighed the females to get their weight after breeding. And then we weighed the offspring and we accounted for the total number of the offspring that was produced. So after the first round of breeding, we exposed them again to this density treatment. We did this so that we can see the prolonged effect of being in a high density environment. And then at the end of all our breeding bouts, we kept these beetles in separate individual cups. We did this so that we can monitor their survivability. So after collecting all my data, I analyzed them using RStudio. The first thing I tested was the cost of reproduction, where I used the two-way ANOVA, and these are the models that I ran. So to determine if there was variation amongst population, I performed a post hoc test to determine if there was a significant difference between populations and their response to treatment. And also I evaluated survival by running a two-way ANOVA, and this was the model I ran for survival. So the first result I will be presenting is weight difference. I used weight difference to analyze the cost of reproduction, and the weight difference was the difference between the weight pre-breeding and weight after breeding. So I observed a very interesting thing here. But before I get into that, HC means high density or high competition, while NC means low density or no competition. So in my first round of breeding, I noticed that contrary to what I was expecting, females from the high density environment gained more weight than females from the low density environment. But here is a very interesting thing. When I analyzed the second round of breeding, I saw the opposite result. That is, female from the high density environment gained less weight than female from the low density environment. And then I began to ask, like, what could be the difference between breeding attempt 1 and breeding attempt 2? A possible explanation for this could be that they had different reproductive value, and so their response, they had different reproductive values. So this could mean that as they age, their response to density changes. That is, they tend to prioritize their offspring over future reproduction. So they invested more in their current reproduction than in their future reproduction. And this was done because they possibly wanted to optimize their overall reproductive success. So I also analyzed the average weight and the total number of offspring. So remember that I said that beetles or rather individuals in high density environment are characterized by bigger body size. So our results showed the trend of individuals in high density environment having bigger body size. But what I was actually interested in was to see if there was population by treatment interaction. And this would be able to inform me if there have been like loss of plasticity or not. So I did not see any population by treatment interaction when I analyzed this. And also, I did not see any population by treatment interaction when I analyzed the total number of offspring. So that was interesting. So I went ahead to analyze survival, and then so I noticed that females in the high density treatment survived less, or they experienced faster mortality than females in the low density environment. I mean, even though I was expecting this to happen, I actually predicted that this was going to be as a result of reduced fecundity. But then this wasn't as a result of reduced fecundity. My results showed that this wasn't as a result of reduced fecundity. So a possible explanation for this would be that in the high density environment, there was high aggression. So also, I observed when I was doing this experiment, I observed that females in the high density treatment box had injuries, like they had loss of limbs or loss of elytra and stuff. So this could be one of the factors that must have sped up their mortality rates. So I went ahead to analyze body size, and then I discovered that in the high density environment, the body size was highly significant in determining the length of survival. That is, beetles with bigger body size survived more than beetles in smaller body size. So this could mean that the fact that beetles in high density environment always have bigger body size, like in their offspring always have bigger body size. This could be that body size could be an adaptation to competition in the environment, or a response to competition in the environment. So, in all, this experiment highlighted the consequences of being in a high density environment, which comes in form of reduced fitness and reduced future fecundity for the females. And then it also highlights the importance of body size in a competitive environment. So this brings me to my second study, exploring the influence of environmental variation and maternal effect on competitive interactions in berrying beetles. So for this study, I'll be focusing on two populations of berrying beetles. One of them is Port Royal State Park, which is just a small patch of land by the river, and the other population is land between the lake, which is a huge patch of forest. Now, beetles from these two locations have like opposite characteristics, in the sense that beetles from land between the lake are characterized with smaller clutch size and bigger body size. Meanwhile, beetles from Port Royal are characterized by so many larger clutch size and smaller body size. So seeing these two opposing characters or characteristics, and based on my previous research, I saw that body size is a reflection or can be a reflection of how dense an environment is, or reflecting the intensity of competition in environment. So this made me ask questions like, what are the adaptive significance of these traits that we see in these two populations? Like, what is their influence on competition? And also, aside size advantage, is there any other modification that an organism need to survive? In other words, do maternal effects play a role in enhancing an organism's or an individual's survivability? So, if that is the case, I would expect that mothers would modify their offspring phenotypes according to our environmental origin, and then population adapted to competition would be advantageous when competing with same-size individuals from other populations. And then I would expect to see beetles from Port Royal State Park like exhibiting inferiority in contests when their size matched with beetles from land between the lake population. So how did I do this? First of all, I did a cross-fostering experiment. That is, egg produced by one individual was raised by another individual. Why did I do a cross-fostering experiment? I did this to get adults of matching sizes. And then how was I able to do this? Bearing beetles exhibit temporal kin recognition. So as long as I targeted females that laid eggs at the same time, and then I switched the females. So this was the switches that I did. I did LBL to PR switching. Please note that LBL is land between the lake and PR is Port Royal State Park. So I did LBL to PR switch, and then I also cross-fostered between population, and this was to account for the confounding effect of cross-fostering. So after I had successfully cross-fostered these individuals and they had successfully raised their offspring, I performed a 5 experiment with the offspring, with the resulting offspring from the cross-fostering experiment. So I did this by placing 2 beetles in a box. And note that I did this 5 experiment for both sexes, that is a separate for females and a separate one for males. So I placed 2 individuals of the same sex and of opposing population in a box, and this box contained moist soil and mice carcass. But for the male experiment, I also placed a female in the box because male beetles have to compete for females, and this is one of their sexual selective traits. So every 24 hours, I went back to check this box, and anytime I see a particular beetle on the mice carcass, I called it a win. So I called a total win when I saw like an individual on this mice carcass for 3 consecutive days. And at the end of the day, I collected all my data, and then I analyzed them using R. So I employed a chi-square test of giving probabilities to determine if the frequency of wins between the two was statistically significant. And then I employed a generalized linear model to determine the interaction between foster parentage and the winner. So for my linear model, I actually used a binomial model where I assigned a value of 1 to LBL and a value of 0 to PR when they win. So this is the first result I'm presenting. When I did this analysis, I saw that there was a trend showing that beetles from Port Royal won a lot of fights than beetles from land between the leg, which was contrary to what I was expecting, although this result wasn't significant. But the fact that PR1 suggested that the competitive advantage of land between the leg might not be just due to regular genetics. So I went ahead to analyze the effect of foster origin on these beetles. So the first one was the effect of foster origin on the LBL population. So from these results, remember that I said I assigned the value of 0 to PR wins and a value of 1 to LBL wins. So this is for the LBL population. And from here, you can see that when LBL was fostered by a PR1, they had a huge disadvantage in winning fights. Meanwhile, when they were fostered by their own origin, by their own population, it did not affect their chance of winning. Also, for the male fights in the LBL population, even though we saw that there was no significant difference between being fostered by PR and being fostered by LBL, we still saw a trend that when LBL was fostered by PR, it had a slight disadvantage in winning. So when we analyzed that of the PR population for the female, we saw that when PR was fostered by LBL, it gave them an advantage. And when they were fostered by their own origin, it gave them like a disadvantage in winning. And also we saw the same trend for the male PR population. So in all, this guy was fostered by a PR mom, while the other guy was fostered by an LBL mom. So it gave him an advantage over this other guy. So why do we see this effect? A simple explanation for this is maternal effect. Remember that I said that LBL is characterized with smaller clutch size and bigger body size. Therefore, this trait indicates a selective advantage for individuals enhanced with competitive strategies. And these strategies can be transmitted differentially from parents to offspring through a unique rearing tactics. So this means that maternal effect can be an adaptation to competition. And if that is the case, then a possible explanation for the disadvantage that we saw in LBL beetles being fostered by PR could be a mismatch in the rearing strategy. We know that PR is characterized by large clutch size and smaller body size, which could mean that they are adapted to low competitive environment. Therefore, PR mothers might have applied the same rearing strategies to this LBL offspring, thereby intentionally or unintentionally making them ill-equipped for competition. So even though they had like physical resemblance in size to their competitors, LBL exhibited traits of small PR beetles, putting them at a disadvantage. So in all, this study highlights the adaptive nature of maternal effect, where mothers modify the phenotype of their offspring to enhance their survival. And we should note that this adjustment do not only benefit offspring within the maternal environment, but it also potentially influences their future survival into adulthood, especially if they find themselves in an environment that is similar to their maternal origin. So, in conclusion, these two studies that I did highlighted the effect of or the influence of maternal origin in determining competitive outcome. So even though body size plays a role in competitive outcome, there can be other modifications which can shape this competitive interaction, and these modifications could come from maternal effect. So on this note, I would like to acknowledge a few people. I'd like to acknowledge my advisor, Dr. Kyle Benowitz, for taking a chance on me and giving me the opportunity to work in his lab. He was one of the few people that responded to my emails when I was searching for grad school, and then he was really excited when he responded to this email. So he has been a good mentor, and I can say that I've learned a lot from him, especially the ability to pay attention to little details. I also want to commend my committee members for their advice, Dr. Kashna and Dr. Catherine Haas for their advice, and I also want to acknowledge my lab mates, Alina, Lauren, Molly. Thank you for helping me collect beetles. I also want to acknowledge Chess for helping me, sending me cool beetle pictures. And also Jessica Varelo, who was part of our lab. She also helped me collect beetles, especially from Port Royal State Park. And then I want to acknowledge NSF for indirectly sponsoring this thesis. So, thank you. Questions?
Just a little one. So you size match the fibers,
Yes.
but their weights were different? Or were their
No.
weights and size, I mean, so both length, pronotal length and body weight were equal in those size matched?
Yes, they were equal to the nearest, I think 0.05 or 0.1.
So it was both, so the body length is directly related to body mass? Or was the body weight that you were measuring?
No, I measured both, so
Okay.
I size matched them according to both.
Okay,
Yes.
awesome, okay, thank you.
As best as possible.
Yeah,
And
no,
that's
I
why there's
understand
still
that. No, I
variation
understand
on
that.
that.
Yeah, of
Exactly.
course, of course, of course, of course. Okay, that's great, thank you.
Just out of curiosity, I don't know anything about beetles. I want to know beetles' social or their solitary. And did you consider if this was going to affect them? Because I know you said in one of the treatment group you had just one beetle, and in the other treatment group you had 10 beetles, right? I
Yeah.
think solitary organisms, would that have like affected like
I actually did not consider that, and I am not sure, but I think they are probably
do they like living together?
no.
Okay.
I think they are solitary.
Okay.
Related to that question, you know, we all know that parent beetles are well studied in their bi-parental care context because male also play an important role in care. So I'm just curious, for your first study, no computation, high computation treatment, why did you focus on the females instead of like bi-parental context? And also, if your study is like under bi-parental context, do you expect to see the same trend? Because now the female has their social partner to help on the current reproduction. So what do you think about that?
Well, even though these parent beetles like can exhibit bi-parental care, the females provide more care for the offspring than the males. So if a male has to be there, like be in the treatment, then I probably would not see this particular effect that I was looking for, because they would have like taken a bulk of the parental care. Yeah, and we probably wouldn't have seen this effect. So I just wanted to like measure the effect that it has on the females, because I mean, in the wild, it's possible that a particular female might not have a male helper. Yeah, so what could be the cost of that, of just her raising that offspring?
One more. So like I know you measure pre-breeding and then post-breeding, and you did breeding two rounds of breeding, right? Did you look at like the weight difference between round 1 versus round 2 treatments, of like how much the females are eating and things like that?
Yeah, that was what I measured here. I mean, that was the result I presented here.
I think she's asking about the weight gain between
Between the
end of
treatments.
route 1 and before route 2.
Yeah, well, the weight gain between the end of round 1 and before round 2 actually served as the initial weight for the females
So you don't know,
for the second round.
but you don't know either like individual beetles gain or lose weight and
Weight
between
in-
treatment.
Between treatment, I think I measured that, but I did not account for it here.
But there's no like significant or anything.
No, there was no significance
Okay,
of that.
That's what I'm wondering.
Yeah, there is no significance of that because even before the second round of breeding, I measured the weight of the females to also like evaluate the weight gain or weight loss after the second round of breeding. So I had an initial weight for it and a final weight after breeding. So that was that. Cool.
So, how would you differentiate between fertility and fecundity? That is my first question. Second
Okay.
question, are there any relationship between fecundity and mortality? And then the third one, in your first analysis, you indicated that you did a post-hoc test to compare the treatment used to see whether significant differences existed.
Mm-hmm.
What specific post-hoc test did you perform?
Okay. To answer your first question, I would assume fertility is the ability for an organism to produce, while fecundity is like measuring their reproductive success or how successful the reproduction is. And then the second question.
Relationship between fecundity and mortality.
Yeah, so as an individual reproduce or as the amount of times an individual reproduce could confer like a fitness cost in terms of reduced fecundity. And also reduced fecundity means like this individual has lost like energy or something. You get? So this can also like indirectly affect or directly affect the rate of senescence. That it can lead to cellular death, like the process of senescence. So that's like the link between the two. And your last question.
Post-hoc test.
Hold on. I'm sorry, I have this at the back of my mind, but I just can't remember. Sorry. I won't be answering your question.
One last question. What did you during this? I mean, these seem like really interesting animals, and I was just wondering, just aside from the research you did, what was something that you walked away with after studying this animal? What were some thoughts you walked away with after? What something new about this animal? What was either what do you like about them? What do you not like about them? What is sort of your general feeling about their general biology that you learned that's going to stick with you about this species?
Well, one thing I like about them was the fact that they actually provide parental care, and because I mean we don't see this in a lot of vertebrate or yeah. So one of the things that I actually took with me from these results or from this experiment, it's the fact that there is maternal investment, and maternal investment actually like plays a role in competitive abilities. And before this experiment, I actually thought that in competition we probably should just consider body size, because there are a lot of studies that have told us about like how body size influence competitive abilities. So when I saw that there is like an effect of maternal origin, I think that was like really fascinating, and I think in the future I would like to like do more studies on maternal effects and stuff.
questions? Okay, thank you so much.
It's actually a wall.